Well, it's certainly very, very good to be back. Very thankful for being invited. Also need to pass along greetings from Lori Seaver, who's with me now in Grand Rapids. She was disappointed she couldn't join me here, but I bring her greetings. So in Michigan, just in case you're curious, people talk about where they're from by looking at their hand. So if you look at your right palm, Grand Rapids is sort of where like your crease, almost the medial side, right where your fourth and fifth digits are. So that's where I come from on the hand. So what I'd like to do is just kind of give a brief overview of the South Carolina Neurotooth Defect Program and use that as a springboard to some new thoughts as far as other birth defects. And I like the idea of birth defects as an example of some of the things that have already come up this morning. And I think of it somewhat as almost like a reverse telescope, in that we sort of identify or notice something, and then we try to refine that more and more to get to why did this thing happen, etc. And I think that works really in all of genetics in a beautiful way. So my interaction with this starts in this state, but certainly South Carolina has a long history with neurotooth defects, in that the southeast United States, with South Carolina as almost the epicenter, has been very much a hotspot for neurotooth defects since they started to become, since they were tracked. I think the beautiful thing for South Carolina is that through Jane and the team, there is a phenomenal program for surveillance and prevention that gives us an incredible amount of data to mine through to actually, again, kind of go down that telescopic route to understanding neurotooth defects better and then translating that to treatment down the road. So these are the numbers over the nearly 25 years of surveillance in the state of South Carolina. And you'll see the black here is the Detoll Rate, and then we divide them up into the three sort of main subtypes, where we have spina bifida here, we have anencephaly, and encephalocele. And one of the things I want you to notice is generally, yes, the numbers have come down dramatically, now basically down to the national level. I think it's important to see that these numbers have not come down to zero. And if you look at kind of just the general trends, it does not look like these are headed towards zero at any point in the future. When you look at the subtypes as well, you'll see, again, decreases, particularly in these two, but here with the encephalocele, not so much a major change in the numbers. This is a map of the state, giving you a sense that over the years of surveillance, really this is an issue that has spread out across the entire state. Then we can extend certainly to the rest of the United States and to the world. One of the challenges with neurotooth defects has been some of the environmental factors, because certainly within the state of South Carolina, there's not necessarily a specific area outside of just higher populations that would be an area to look at in a little bit more detail. But when we expand across the globe, we get into some areas where the rates are certainly higher, and you think about some of the nutritional things that were even brought up earlier this morning. So coming back to this chart, this is where we want to sort of slice and dice these numbers as many ways as we can to try to dive a little deeper into what we can learn about this. And so let's start with, I think, probably one of the easiest ways to break this down, and that's taking straight up neurotooth defects that are isolated versus neurotooth defects that have other associated findings with it. That's definitely going to be a bit of a generalization, because when you talk about other associated findings, the list of that is quite long. But again, if we think about how we're trying to narrow this, what's the utility of looking at this group, sort of those isolated cases? How can we understand neurotooth defects more from those specific cases? What about the ones with other associated findings? Does this help us understand various syndromes, chromosome differences, etc., that again would help us understand neurotooth defects a little bit more? So if we break down the three subtypes of neurotooth defects, you'll see these are the numbers for the other associated findings. So the sort of taking this chunk and then slicing it into three. I think it's interesting to see that spina bifida and anencephaly come a bit closer to this number than the encephalocele number. It's going to be a little harder because of the numbers in general, the total numbers of cases that we're dealing with. But I think this gets at the root of some of the debates in the NTD community as far as the mechanisms for these. Can we lump all of these together? Some people would say no, others would disagree. But again, we think about neurotooth defects with the neural tube just not closing, and you know the sort of the two ends of it. But how do we understand this a little bit better? So when we look at those other associated findings, so the smaller piece of the pie in the previous slide, this is what it comes down to. And I think as I've sat through some of the talks this morning, it's interesting that it feels like all the pie charts that we're showing, there's half that's we don't know, which is, I don't know, that is kind of clinical genetics, right? Half the time, good luck, we don't know. That's why I grew this beard, so I could stroke it and try to look like I was thinking, and I have no clue what's going on. But again, when we slice this up, you'll see a reasonable chunk for chromosome differences. You'll see some of these syndromes have neural tube defects as necessarily a part of them, but maybe others down here that you wouldn't necessarily think of neural tube defects as being a specific piece of that syndrome. So we talked about sort of occurrence and learning information from that, but I think one of the marvelous pieces of tracking this over so many years is recurrence, and taking those again as another level of further detail to try to mine it. So these are sort of, this is the flow chart over 25 years of the over 1,200 cases of NTDs that have been tracked. Of those, there have been 663 subsequent pregnancies to these families. And you'll see that we can divide these up into women that were on high-dose folic acid and not during those subsequent pregnancies. Again, one of the parts of the South Carolina program that's so wonderful is providing those families with access to the folic acid that they might otherwise have had a little bit more difficulty obtaining. And I want you to look at these numbers here. So when there's high-dose folic acid, you'll see the recurrence rate is basically, let's say 5 in 500 basically. So that's a 1 in 100 recurrence risk. If you look at the nearly 100 pregnancies where there was not folic acid use, you'll see that it's about a 1 in 10 chance. So you go from a 1% to a 10% chance. That's a tenfold difference. So again, speaking to the power of folic acid usage to prevent neural tube defects, but then saying, hey, well, it doesn't prevent every case of neural tube defects, so there suggests there's something to be learned there. And then I think one of the other things that I was going to say, I don't remember, so we're going to move on. So, those recurrences on folic acid. So those 5 cases where mom was on folic acid for a subsequent pregnancy and there was another child with a neural tube defect. I think one of the powerful things is seeing that generally those 5 cases have an explanation for them. And that, I think, for me, is kind of the tip of that telescope, where we've taken it down to the highest yield cases and been able to understand why. These 2 genes here are part of a recently accepted GGC publication that will be coming out, basically describing the first couple of genes that really explain neural tube defects. I can remember the day that the APAF1 gene came down from the lab. I had to bring my daughter into work that morning, and my phone was ringing off the hook. It was absolutely insane, but it will stick in my head forever. APAF1. All right, so one of the other projects that we worked on using this data was to try to characterize what we tried to be provocative and call folic acid failures. And this was actually published in AJMG a couple of years ago. And in general, looking at those women that, again, took folic acid, whether it was the first time for a neural tube defect or a recurrence, and trying to look at what we could characterize about those that was a little bit more unique. We found older mothers, which always sort of maybe rings a bell in genetics a little bit. We found some differences in ethnicity, which again has been known before with neural tube defect rates. That the majority were isolated, again, kind of getting to those genetic factors versus environmental factors, but then specifically, again, spina bifida stood out with other associated anomalies. So again, kind of going back to that, what's the mechanism for the various neural tube defects? So I mentioned previously the South Carolina program, and again, education, trying to prevent occurrence, certainly recurrence as well, but occurrence in general, and just trying to get the message out. And this was alluded to a little bit earlier, that yes, even when we get the message out, it's not always received and acted upon. So this is a telephone study that GGC has been a part of for many years that recently had an update. And you'll see the percentage of women that were even aware of folic acid being a benefit still never touching 70%. And then when you look at the women of childbearing age who were taking folic acid, you'll see there's a gap there. And again, these numbers never even approach 50%. And these are the two numbers from the 2017 survey. I believe you've heard relatively recently about a Zika telephone study as well. And I was struck thinking about this and the Zika numbers is that Zika is kind of what I'll call maybe like the first millennial teratogen, birth defect, etc., that's sort of been in like the social media age. And the numbers that were found that women knew about Zika far more from the media, from social media, that sort of stuff, rather than healthcare providers. And the numbers with folic acid are, again, mostly reliant upon healthcare providers and more general education. So I think that's an interesting thought for the future. How do we get our message across about these sorts of things in a way that people are willing to listen to, let alone women who are of childbearing age now and even into the future? How are they going to receive information? And I think in general, this is sort of the payoff with the program is the theoretically saved cases of neural tube defects. So this is sort of projected, and again, these numbers aren't completely updated, but getting to nearly 1,000 cases of neural tube defects within South Carolina that have been prevented because of this program. And someone mentioned earlier the cost associated with each case of neural tube defect. So take that 1,000 and multiply it by that, tens of thousands of dollars, we start to add up to some pretty significant cost savings based on educating the public. All right, so I want to shift a little bit here to talk about sort of a similar case. So this is one of my current partners, Dr. Paul Mark, and he has been involved in a recent paper in the New England Journal about birth defects, niacin supplementation, and NAD or NAD deficiency. So the story for him starts with this particular patient that he saw for a variety of health concerns. And if you look down this list, you'll see a number of things that have been seen before, or at least talked about with bacterial association. And just to remind you, that's an association, so things that are seen more together necessarily than a syndrome. But if you look at her picture here, she looks a little bit more distinctive than maybe someone with other bacterial patients that might have been seen, specifically sort of looking at proportions in her stature. So he did whole exome sequencing on her and found compound heterozygous mutations in a gene called KYNU, or K-Y-N-U. And through a match with a partner, connected with some folks in Australia to look at its function in this pathway. So this is the NAD synthesis pathway, nicotinamide adenine dinucleotide. And some of these compounds might look familiar to some of the audience here. And I'll point you to the KYNU gene and also the HAO gene, which is right next to it. So again, the thought being here with this particular pathway, when this gene is mutated, it blocks it and prevents synthesis of NAD. So this just gives you a sense of the levels of seen before and after blocks in that pathway. And I'll just point you over here so you can see where the block is. So you're seeing high levels before. This is one from one of the HAO gene patients. And you'll see afterwards all these levels are significantly lower. The 4 patients that they presented in the article all had features consistent with bacterial association, but I'll remind you that bacterial association has a bunch of different things that go with it, so the clinical criteria is a bit vague. So the thing that sort of sealed this a bit was that there's a mouse model for mutations in this gene where the mice have features of bacterial association. So these are the mice that I would have you look at. So there's the KYNU and the HAO, and you'll see vertebral differences, pretty significant ones. These are the hearts right here, so they're pointing out heart defects. These are mouse kidneys, so comparing it to unaffected, you'll see the size difference there. These are the limbs, looking at limb differences, and then they also noted clefting here as well. So we have a mouse model where these genes are affected that have basically all of the clinical features of bacterial association. Now we have a few patients who have the clinical features of bacterial association. Can we make a connection here, specifically with the niacin and the NAD pathway? And the exciting thing from the papers, particularly with the mouse model, was if you give the mice enough niacin, it basically recovers or saves them. So the idea of thinking about birth defects, thinking about what causes them, and then jumping to treatment is always incredibly exciting to think about. And so the question that this sort of raises and that I would leave the audience with is, with this particular pathway, have we in some ways stumbled upon an analog to our neural tube defects with our folic acid helping to prevent them? Because the thought is, have we just basically discovered two genes that are involved in this pathway? When they don't work, these levels just happen to be off, so that's it? But I would suggest to you that what about the idea of bacterial association as more of a general birth defect? Because we know other genetic syndromes look like bacterial association, so say something like Fanconi anemia. It would be interesting to know what their NAD levels were. The other thing that I would say to you is, what about maternal diabetes? The effect that maternal diabetes can have on children gives you a lot of the findings that are seen with bacterial association. So was there something about mom's ability to metabolize niacin or her NAD levels that you could theoretically treat pregnant women or women with maternal diabetes with extra niacin to prevent birth defects? So it's an interesting thought, and one that my partner is giving a good bit of consideration right now. One of the challenges that has come with it, though, is that this is how the media reacted to this article being published, was basically to put it in a very black and white context that, well, hey, we discovered why birth defects happen, just take a little bit more niacin and everything will be fine. And clearly things are not as simple as that, but again, these are some of the challenges that we face, again, with communicating these issues as time goes on. All right, so future directions for this, I think, are many. As Paul and I talked about this, we talked about, well, who would be a reasonable partner to work with on this? And so that's where the Greenwood Genetics Center has come in. So Paul has had some meetings and had some discussions, and it's kind of exciting to think where this may head in the future. So lastly, I want to remind you about a proposal that I made about 3 years ago when I gave my seminal case review about genetics in space. I had suggested a Greenwood Genetics Center space program. I'm assuming that's what the afternoon sessions are about, an update on how you've been doing. And so I had told you that I was doing my part back then to start training our first astronauts. So this is 3 years ago, and I wanted to show you that we are still hard at work on this, and the Bupp family. So we now have 2 future astronauts for you. So with that, I think it's always important to remember that genetics is very much a team game. This is the current team that I have in Grand Rapids, Michigan. And I will show you these pictures because I kind of had this idea, and you can decide whether this sounds familiar, that every summer we take an afternoon and we go and we dress up and we play ridiculous games. And then we eat food together and have a lovely time. I have yet to call it Fun Day, but in all seriousness, as I have reflected on how I have tried to set up my career at this point, I see complete connections back to my time here. As far as we do a monthly case review now, we do our Fun Day, and I'm just incredibly grateful for everyone in this room and for inviting me here to talk today.